The cell cytoplasm consists of the ectoplasm and endoplasm. Ectoplasm is just that very fine piece of cytoplasm immediately underneath the cell membrane. It doesn't contain any organelles. All it does is contain very fine actin filaments, and those actin filaments help to support the very delicate cell membrane. The endoplasm is that part of the cytoplasm that contains all the organelles, those structures that are membrane bound and that serve specific functions in the cell. The endoplasm also includes what we call cytoplasmic inclusions. Cytoplasmic inclusions are structures that may not be in all cells. They're inclusions that might be peculiar to individual cell populations, and we'll see those when I describe these in more detail in a later lecture. And of course, there is the cytosol, or the cell sap, or cell fluid. One important fact to remember is that the eukaryote can divide by mitosis, or if the cell is involved with becoming a germ cell, for instance spermatozoa in the male, or an ovum as the female gamete, then those cells are produced by a process called meiosis. What I've tried to show you in the last couple of lectures is just what the concept of what a mammalian cell is, and that the mammalian cell contains a nucleus with a nucleolus and cytoplasm bound by a cell membrane. You can see in the central part of this image a chain of nuclei, rather a condensed lot of little circular profiles. They're the nuclei of cells. The clumpy stained material you see is chromatin. There's other nuclei and cells in the periphery, but I want you to focus roughly on this structure running down the middle. It's a kidney tubule. And now, hopefully, you can identify a nucleus. And around that nucleus is a very, very pale stained pink region. That's the cell cytoplasm. Then if you look very, very carefully towards the center of this tubule, I'm sure you're able to see very, very fine little pink lines. That's the cell boundary, the boundary between one cell and its neighbor. And if you look at those little lines, roughly, you can work out that the cell shapes are almost cuboidal. They're cuboidal shaped cells, and that's also indicated by the fact that the nuclei are circular. If they're columnar or tall cells, the nuclei would tend to be elongated. Now you can see details of these cells because you use a tool in histology called a microscope. And a microscope does two things. It magnifies or zooms in on a slide of your tissue, such as the kidney here, and it also provides you detail of what's on that slide or in that section of kidney. And first of all, you need to cut a very thin section of the kidney to be able to magnify it and also see the detail. So what I really mean is to look at all the histological sections of body tissues and organs. You need to use a microscope, and that's going to magnify what's in the section, and it's going to provide detail. It has the resolving power to distinguish objects that are very, very close together. With our eyes, we can only distinguish objects that are about 0.2 millimeters apart. We can't see any objects that are closer than 0.2 millimeters or 200 micrometers apart. The light microscope enables you to look at the detail you see in this section, and you can see objects that are as close as 0.2 micrometers apart. Again, that's what I mean by greater resolving power using a microscope. I'll mention resolving power again in a later lecture. So now you have enough information to hopefully extend your knowledge a little bit further and look at some specific cell structures in more detail, namely organelles and inclusions.